The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or to view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
All right, so today then we are going to talk about eye movement control. This is a fact-laden set of topics. We're going to talk about the basics of eye movements, and we're then going to talk about the various neural structures that are involved in eye movement control. The interesting thing about studying eye movement control here, which is very closely obviously related to vision, it's also very closely related to the oculomotor system because it involves moving the eyes. Now, the basic fact is that we move our eyes practically endlessly every day. We make about 3 saccades a second, and we make more than 150,000 saccades every day, and it's not something we ever think about. Yet, every time you look at something, you have to decide as to where you're going to look next. And you're not even aware of it. It just happens automatically. It's a remarkable system, works with incredible rapidity, and it involves recognizing objects out there, recognizing the visual scene, making a selection, moving your eye there, and keep doing it, as I said, 3 times a second. So this is obviously the eye movements that you make like that, jumping from one location to another. And with each of those eye movements, as I've said, you need to decide where are you going to look next. So that's quite an amazing feat. And it's not just something that we do as humans, but it's also done regularly by animals. And most of the work I will tell you about has been conducted actually on monkeys whose eye movements are remarkably similar to those of our eye movements. Now, let's first of all go over what I'm going to cover in the next 2 sessions. First of all, before I do that, I just thought I'll show you an amusing cartoon from the New Yorker, which was made in 2001, which sort of puts in a nutshell what the nature of all this is. Here's a cat. It's going to try to jump up there to get that thing. And so it has to make a decision where that thing is in space, has to make the calculations as to how to generate the motor activities to jump up there. Now, when it comes to the eye movements, then you don't have to jump up there, but you have to decide how to jump your eye from one location to the next. So what we are going to try to understand is what are these calculations that we perform to be able to make accurate eye movements from one location to another, and what neural structures are involved in this, and what are the various rules these neural structures are involved in to generate those eye movements. Now, here are the topics. First of all, we're going to look at just the basics of eye movements. Then we are going to look at the so-called eye plant and the brainstem nuclei, which are involved in moving your eye muscles. Then we are going to look at an important structure in eye movement generation, which is called the Superior Colliculus, that we hadn't talked about yet. And then we are going to look at the visual inputs for saccade generation. We're going to examine what the various types of retinal ganglion cells do to be involved in eye movement generation. In particular, we're going to look at the midget and the parasol cells. Then, perhaps a little bit today, but most of that next time, we're going to look at the cortical structures involved in eye movement control. Then we're going to look at the effects of paired electrical and visual stimulation, because that will give us additional insights about the nature of eye movements. And then we're going to look at what happens when you have various deficits in motor control as a result of having lesions in various parts of the visual and oculomotor systems. And then we are going to look at some pharmacological studies to try to understand what the pharmacology is of eye movement control. And I should always say visually guided eye movement control. All right, so let's first start then with the basics of eye movements. We can ask the question, why do we move our eyes? Now you already have the answer to the first reason we do that, which is that in the retina we have a highly specialized system in higher mammals and in humans, where there's only a very small area where you have very high density of photoreceptors. And so to see fine detail, you need to move your eye to the location to be able to analyze it at a high level of acuity. So that then involves the ability to acquire objects for central viewing. All right? Because it's a central viewing that allows you to have high acuity. Now, those are accomplished predominantly by saccadic eye movements, just like the ones I had mimicked at the very beginning. You make these very rapid eye movements from one location to another. Now then, another important part of eye movement control is that when either you are in motion or whatever you're looking at, like a bird flying in the sky, to be able to analyze it, you track that object, all right? You make what is called smooth pursuit eye movements, okay? So that is another important mechanism that enables you to analyze things accurately in the world. Now, yet another factor is that when we move about, it's important for us to be able to, for our eyes to be stable with respect to the world out there. And one of the mechanisms involved in that, as we shall see, is the accessory optic system, and that's what you're going to be writing a paper about, that is involved in controlling the eyes, involving both visual stimuli and the vestibular system. So that is what we're going to talk about. Actually, that part we're probably going to talk about when we, in the next to last lecture, not next time, but time after, when we talk about the accessory optic system a bit. All right, so now then, just to reiterate then, we're talking about the vestibular reflex and the accessory optic system. All right, so now then, people have classified eye movements into some very basic types. We have so-called conjugate eye movements, when the two eyes move in unison. And then we have vergence eye movements. So when an object comes close to you and goes away from you, then the two eyes converge or diverge. And that still has to be done to make sure that the images you see in the world there fall upon corresponding regions of the two eyes. Now the conjugate eye movements fall into two basic categories. They're called saccadic eye movements, the ones we had talked about already a bit, and smooth pursuit eye movements that causes us to be able to track objects so that we can keep that object on the fovea. All right? Now, vergence eye movements, you go back to that picture I'd shown you before, in which you have the horopter, or the Withmuller circle, if you remember. If any object is along the Withmuller circle, like going from 1 to 2, say an object jumps from here to here, then when the eyes track it, they fall on corresponding points into two retinae. However, if there's an object that falls outside or inside the Withmuller circle, as shown here, then the second object falls on non-corresponding points, and so therefore you have to bring about eye movements that involve vergence, either divergence or convergence. For the most part, today especially, and next time as well, we're going to be talking about saccadic eye movements, and then, as I've said, after we have covered movement, we're going to look at eye movements that involve pursuit and vergence. All right, so let's now take a look at a human subject and see the eye movements the subject makes. I mean, you're perfectly aware of it, but I thought it would be fun for you to look at it. So here's a subject and is looking at something, right? And all these eye movements, obviously there's nothing moving out there, so these are all saccadic eye movements. The head is fixed. And so now once you've seen this, there are a number of questions that comes into one's mind, okay? The first one on the not too scientific end is, well, was that a male or a female? Or secondly, what was that person looking at? And then thirdly, way down the list, you say, well, how does the brain do this? Okay? So let's see if we can answer these three questions. The third one will take the 2 lectures. But the first and second one can be answered fairly quickly. Okay, was it a male or a female? Anybody want to commit themselves? Huh? Male, male, male. Anybody think it was a female? Oh, you guys are good. It was a male. Very good. All right, now the second thing, of course, is what on earth was this person looking at? Now, there is no way you could glean that until I tell you, right? So let me tell you what this person is looking at. And what this person is looking at is a picture which is created by René Magritte and actually resides at the National Gallery of Art in Washington, D.C. It's quite an interesting painting, and that's why I'm showing it to you. And it relates closely to perception, the curious aspects of it. And you can see this doesn't make sense quite, right? But it's because of that, because of the puzzling nature of this picture, that it is in a fabulous museum, because it is so unusual and different and makes you think. So anyway, what happens then, suppose you started here when the picture came on, then you're going to have to make a decision where you're going to look next. So what we're going to look at next, most commonly you would look at the eyes of the rider, then you would look at the eyes of the horse, okay, and so on, look at various outstanding features. Now the way this looks more or less in real time looks like this. So that's what this person was looking at, and that makes you aware of the fact that whenever you look at a picture, you look at anything, you make all these eye movements in this very short time for you to comprehend the scene and to understand it in some detail. Now, when this kind of eye movement series is made, you wonder, is this something that animals also do? And the answer is yes. Monkeys certainly do, most mammals do, and even birds do this kind of stuff. So what I want to show you next is, first of all, what the pattern of eye movements is in humans, and who the initial person who did beautiful work on this, whose name is Yarbus. All right? Now this is a famous sculpture, the so-called bust of Nefertiti. And what Yarbus did, he looked at the kinds of eye movements a person makes when they look at this figure to comprehend it. And you can see what is interesting about this. A lot of saccades along the edges, and when there's a lot of detail, and not too many saccades made to regions where there's smooth areas. So what Yarbus did, he actually published a book on this, analyzing the nature of our eye movements, just looking at it behaviorally like this. Now, if you do the same thing with monkeys, I'm going to show you that monkeys, here's a bunch of monkeys, they also move their eyes just like we do. They also have foveas, of course, as we had discussed. And so as they move around and look around in the world, they make even more eye movements than we do. They're a little bit more quick about it. And so they make many, many eye movements and make about maybe almost 4 saccades a second. Now, to see whether they also actually select objects in a sensible way, what can you do is you can present a bunch of displays. Here's a bunch of round ones, okay, I mean, in around locations, I should say, different objects. And here we have a bunch of them is arranged in a rectangular fashion. Then on the right, with the head fixed again, you see the kinds of eye movements the monkey makes. And what you can readily do, even if you didn't see these here, you can tell that there must be a whole bunch of objects at these locations, including one in the center, and here that the objects must be aligned the way they are there. So clearly the monkeys do the same thing we do. They tend to look at particular objects in the scene and make accurate saccades to them to be able to analyze them. So because of that, it is natural to take monkeys and to study their eye movements and to study the underlying neural mechanisms in these animals, which will then make us more capable of understanding just how the brain moves your eyes about. All right, so now let us become more concerned with the neural mechanisms and the machinery involved in eye movement generation. And so we're going to refer to this as the eye plant and the brainstem nuclei, which are in the midbrain and lower portions of the brain, not in the cortex. We'll talk about the cortical factors later on. So let's start, first of all, with the eye muscles. Each eye has 6 extraocular muscles, as they are delineated here. They're called the superior and inferior recti, the medial and lateral recti, and the obliques, the superior and the inferior oblique muscles. Now, the remarkable feature about these muscles that renders them different from the muscles that you have in the rest of your body is that the fibers of each muscle run the entire length of the muscle itself. It's not segmented. Most muscles in the body are segmented. Because of that, they often are difficult to understand the nature of their operation. But here we have a situation that since all the fibers run the entire length, it is relatively easy to comprehend the basic mechanisms that generate the eye movements at this low level. Now, the next thing that's important to know is that the eye is sort of a balanced structure. It doesn't have any weights anywhere, all right? And so when you move the eyes, it's not like that you have to pick up an object. If you were to pick up a heavy object and you thought it was a feather, then it would practically hit yourself in the face. Okay? But if you know what the object is, you can correct for the way you're going to lift it, all right? Which adds another huge dimension on how you move your body about and how you operate your muscles. Luckily, when it comes to the eyes, that is not a factor, and because of that, it's easier to understand the way it operates. All right, so now that we have this, the next question one can ask is, how are these muscles innervated? I mean, obviously you have to have some nerves that connect to it that cause the muscles to contract or to let go. The way that works is that the nerves that connect to the muscles of the eye release acetylcholine that causes the muscle to contract. Okay? So that's the basic, that happens everywhere in the body. So that's very basic, you already know all that. But what you probably don't know yet is what are the neurons, where do they come from, in other words, to innervate these muscles? Well, it turns out that there are 3 cranial nerves. How many cranial nerves are there in the brain? 12. Okay? 3 of those, believe it or not, are involved in the control of eye movements. And so we can designate that. We can tell you that the lateral rectus, okay, is innervated by the abducens nerve. The abducens nerve comes from the abducens nucleus. Okay? Now, the superior oblique muscle is innervated by the trochlear nerve, which is the fourth cranial nerve, okay? And the rest of them are innervated by subnuclei of the third nerve, okay, all right, which is called the oculomotor nerve. So you have these three. Oculomotor is the third, trochlear is the fourth, and abducens is the sixth. So it's these three nuclei, with note again that the oculomotor has subnuclei that innervate these muscles. All right? So now that we know that, the next question is, how do these neurons from these three nuclei act to do this? But before I tell you that, I want to make sure that you learn what the 12 cranial nuclei are. What you can do here, and I'm using a so-called clean mnemonic device, on all the Olympus towering top, a fat-armed girl vends snowy hops. That's easy enough to remember, right? The first letter of each designates each of the 12 cranial nerves. So if we then look at that, here they are. All right, the first one is the olfactory one. The second is the optic. Then we have the ones in blue here, which innervate the eye muscles, oculomotor, trochlear, and abducens. All right, and then one of the important ones that you're going to hear a lot about is your auditory one. That's number 8. All right, so those are your 12 cranial nerves, and then you don't have to remember this. It might help to know that, of course, you have a whole bunch of spinal nerves as well. You have 31 of them, okay, and those 31, of course, each doubles, okay. One on the left side, one on the right side. Okay, you have 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. All right? Now, you don't, as I say, you don't have to remember this, and I'm not even telling you have to remember these, but it's good to know that. It's something that you can commit to memory, so that when you go to a party and talk to somebody, you can say, hey, you know about the 12 cranial nerves? And they can say, see you later. All right, so anyway, we can move on now and see how these neurons that innervate the muscles, that's often referred to as a final common path, okay, how they operate. Actually, that underwent quite a bit of debate for a long, long time. I'm not going to go into the debate itself, but I'm just going to tell you how it works. But the fact is that these neurons are involved in generating all sorts of all the eye movements we talk about, meaning smooth pursuit, meaning maintained eye position, and saccades. And so here's an example. This shows vertical eye movements, just the vertical eye movements, okay? And what you can see here is that the more the eye looks down, like here, the more the sustained activity to keep the muscle contracted, to keep the eye down. Now, but you also have to remember that while this is happening here, the opposite muscle on top, the superior rectus, has to let go. So there would be no activity to contract the muscle in the superior rectus, not muscle fibers in the superior rectus. Now, secondly, the important thing to notice here is that whenever there's a saccade, and so on, when there's a little saccade, there's a high-frequency burst. And the high-frequency burst is short. When you have a big saccade, there's a much longer high-frequency burst. And when there's a saccade in the opposite direction, there's a pause. So what do you think happens when there's a pause? In that case, the superior rectus would get a burst and would contract rapidly to get an upward eye movement. So this muscle will cause your a downward eye movement, and the superior rectus records an upward eye movement, of course. Okay? So that is the basic layout. Now this is schematic, but to show you that this is real, I'm going to show you some data here of a monkey doing the actual eye movements, recording from the oculomotor nucleus, and in this case again innervating the inferior rectus. And what you can see here are a bunch of saccades, and here the monkey is tracking an apple as it moved down. Okay? And what you can see, let's look first at this side, at the bottom. What you can see here that somewhere along the line here, as the monkey is tracking, this particular neuron begins to cut in, and then gradually it increases the rate of its activity, as a result of which you get smooth pursuit eye movements. Now if you look at the upper part, once again, as I had shown you, the frequency of activity is proportional to the angular deviation of the eye, in this case because you're talking about the inferior rectus in the vertical dimension. And whenever there's a saccade downward, there's a high frequency burst, and whenever there's a saccade upward, there's a pause. All right? So that is the basic nature of this. And what is so lovely about this is like a machine. This is so lovely that if you then collect data from several cells, and here's an example of it, and you look at the number of spikes per second and the angular deviation of the eye, it shows you that each of these 4 neurons acts in a linear fashion. As the eye deviates more and more, the frequency of activity gradually increases. This is not saccades, obviously. This is maintained activity. Let me go back again, like here, like here, like here, and like here. Okay? So you get this beautiful linear function, which again, as I've said, makes it relatively easy to understand quantitatively the process of moving the eyes. So that is the basic mechanism that you see in the oculomotor nuclei. And the important thing to understand then is that the activity of these neurons is involved both in making saccades and in maintaining various eye positions. The reason I emphasize this, because maybe about 30 years ago, there was a big debate. Some people argued, when they initially began to record from these oculomotor nuclei, that we have 2 different sets of neurons in the final common path, one of which controls saccades and one of which controls maintained position or smooth pursuit. Well, that's not the case. The case is that these neurons do both. Now, why did this confusion arise? Well, it arose because it was found that if you looked at the so-called supranuclear complex, which are neurons which are sort of above, if you will, the oculomotor, trochlear, and abducens nuclei, there are a whole bunch of subnuclei there. We're not going to go into details about them. You don't have to know that. But at any rate, in those nuclei, there are neurons that are indeed separately coding different attributes. And I'm going to show this. But before I do, I want to make one more important point. If you take a microelectrode and you put it in one of these nuclei that controls the final common path, if you stimulate there at a high frequency, in this case 500 hertz, if you increase the duration of the stimulation, you get progressively bigger saccades, which proves with the stimulation that it's indeed the duration of the high frequency burst that defines the size of the saccade that you elicit. All right, so now we can move on, and in this case, as I say, you don't have to know this, but I want to just mention it briefly. We have here's the eye muscle, and then we have, in addition to the final common path, we have a supranuclear complex in which have various neurons. There are some which pause, in other words, they don't code saccades, and some which fire only to saccades. I think that's the only ones I need to point out. But at that level, they're separate. But once they come together in the oculomotor complex, then they impinge on these neurons of the final common path that carry both of these signals. All right, so that then is the essence of the oculomotor complex that I want to cover. And so we can have sort of a quick summary diagram here. We're going to make a diagram that's going to grow over this session and the next one. And it's going to be very complex in the end, unfortunately. But here we have the brainstem oculomotor complex. And the way to put this, because we talk about what kinds of coding operations are involved, this carries what we're going to call a rate code. The higher the rate, the greater the angular deviation of the eye. And of course, the longer the high frequency burst, the bigger the saccade. So that is the so-called rate code. So this is the basic layout then. And of course, if there's a lot of activity here, this muscle is going to contract, the eye is going to deviate downward. At the same time, you're going to get a signal to the upper part that's going to let go. In other words, the signal is going to be terminated so that you have no activity in the upper part so that that muscle can expand. So that's the basic layout at this point. And as I say, this will grow and grow and grow and grow until by the end of next time, it's going to be really complicated. But that's how the brain works. It's complicated. All right. So with that, then, we are next going to move to the so-called superior colliculus. Now, the first thing I want to say about this structure is that this region of the brain, I'll show you a picture of it in just a minute, is one that has undergone tremendous changes in the course of evolution. In more primitive animals, like in fish and in amphibia, what you find is that this structure is the prime structure for analyzing vision. So it does vision and then generates commands of various sorts, including commands to make eye movements or generate other kinds of movements in different animals. And this is the case because in these animals, there is very little cortex. But with the process of encephalization, more and more of the analytical undertakings the brain was involved in had been relegated to the cortex. And that also meant that more and more of the way we analyze vision became more central, as we had discussed already, with cortical mechanisms. Now, to make this clear, let me show you three drawings of a so-called toad, a rabbit, and a monkey. And what you see here, the toad, here's the superior colliculus. This structure is quite large relative to the rest of the brain. Then, when you come to the rabbit, there is some degree of encephalization, so this structure is much smaller relative to the rest of the brain. And then when it comes to the monkey, it looks like that the superior colliculus is actually quite tiny. And in monkeys and humans, the superior colliculus is very small. It's maybe about 5 or 6 millimeters in diameter, and yet it's an extremely important structure, and we're going to examine just what this structure does. And later on, we're going to examine what happens if you lose that structure. All right, so now, here is a sagittal midline section of the monkey. So you can see this in the reel. And here we have the lunate again. This is what part of the brain here? This is the cephalic area? Okay, good. This is area V1 here. All right, then, if you look down here, you see this little bubble-looking thing here? That's the superior colliculus. So that is a structure that we are going to look at in some detail. Now, if one enlarges this is in the cat actually, and one takes a coronal cross-section of the superior colliculus, you can see it's a fairly complicated structure. It has several layers. People have distinguished at least 7 clear layers. And you can see the upper part here is often referred to as superficial gray. Okay? There are 2 layers of that. The very top layer gets an input directly from the retina. the one little further down, I'll elaborate on that in a minute, it gets input from the visual cortex, and then we have a whole bunch of other layers that we are going to examine in some detail in just a minute. So that's the nature of this structure, the superior colliculus. And the fact is that in more primitive animals, actually, it is a structure has many more layers. In some of these more primitive animals, there are as many as 14 layers in this structure because it is heavily involved in eye movement control, as well as in the analysis of vision. Now, the interesting fact about the way this is laid out in the cat and in higher animals and in humans, that most of the cells in the various layers reside in those layers. They don't talk to each other that much, okay? But they get a lot of inputs from many other structures that control those layers. And I'll go into that in some detail in a minute. Okay, now, the next important fact about the superior colliculus, which makes it similar to the lateral geniculate nucleus in the cortex, that the visual field is laid out in a topographic manner in the superior colliculus. This is sort of a top view of the colliculus. This is the visual field. And once again, the rule, if you remember, is that the contralateral half of the visual field is that projects to the superior colliculus. And of course, the opposite then is the case for the other side. And then if you look at the nature of the connections, you find that everything that's close to the fovea, okay, foveal representation, projects to the anterior level of the superior colliculus. So this is sort of the foveal region. Then the upper visual field, okay, projects the medial portion of the colliculus. The horizontal meridian area projects to the horizontal meridian of the colliculus in the back. And the lower part of the visual field projects to the lateral portion of the colliculus. Now, why do I emphasize this? You will see in just a minute why that is important. It's very important for us to understand that there's a lovely topographic layout of the visual field onto the superior colliculus. All right, so now we are going to move on and examine the question of what is the nature of the responses of neurons in the superior colliculus. Now, I can tell you right off that in the monkey colliculus, in the cat colliculus, the nature of the responses of these neurons is not particularly interesting. They're small receptive fields, and they tend to respond both to the onset and the termination of a visual stimulus, meaning that they seem to get an input from both on and off, okay? And then if you make the stimulus progressively larger, you get an increase in responses, but then once you make it quite large, in this case 10 degrees and 20 degrees, the response greatly declines, which says that just like in the retina, these neurons have centers around antagonism, and they respond vigorously to small spots and very little to large spots. Now, that's a basic layout. But then if you ask the question, well, are these cells in the colliculus orientation and direction specific? Are these cells color selective? For the most part, that answer is no. Not entirely, but mostly no. Meaning that these cells are not that really interesting. They're very, very, very basic. So that is the nature of the responses of these neurons in the superior colliculus, in the superficial layers. I've told you that the superficial gray, which consists of two layers, the one that gets a direct input from the retina and the other that gets an input from the cortex, gives you vigorous visual responses. Okay? But now, when you get into the deeper layers of the colliculus, you find something very exciting and extremely interesting. And that is sort of diagrammed here. What we have here is a bunch of eye movements, okay? And then look at this cell. This cell responds, it's the same cell throughout, responds vigorously to a small saccade, okay? And responds well before the saccade is made, okay? That's what happens here. But then if the monkey makes bigger saccades, okay, and some even in the same direction like these here, you don't get a response. So what does that mean? That's curious. So what that means is that these cells, some have something to do with eye movements, all right? This cell fires vigorously and then eye movement ensues. Fires vigorously and eye movement ensues. Now there's an eye movement that ensues here as well, but there's no saccade. So how do we explain this? Well, what you do in these kinds of experiments then is that you collect extensive data to see when a cell like this fires and when it doesn't fire. And you can generate a response curve, if you will, or a response diagram to see when it fires and when it doesn't fire. So now to look at that, here's an example of that. In this case, all the white spots are saccadic eye movements generated from a central point, okay, that were not preceded by saccadic neuronal responses, and the red ones show the times when the neuron you're recording from, the single cell, responded vigorously. So that meant that whenever a monkey made a saccade in this direction, there was vigorous activity in this neuron, meaning the neuron sort of had an important role in generating an eye movement, whereas all the other saccades did not generate any response in this particular neuron. But to make this clear, there are many different neurons in many different parts of the colliculus, and so when you generated the other eye movements, other neurons fired. All right? So how can we analyze this in more detail? Well, the way we can let me just add one more point here. This green circle here is what we're going to call the motor field of this particular neuron. Okay, so that's a new concept for you, a motor field. These neurons in the intermediate and deep layers of the colliculus have motor fields that define the size of the saccade. Now let me say one more thing in anticipation, that if you then study where this cell's visual receptive field is, it's right there relative to the central fixation point. So it means that this particular cell fires when a saccade is made into the receptive field of that neuron. Okay? So how can we verify this interesting, clever arrangement in the superior colliculus? Well, the way we can do this is to, instead of recording, or actually I shouldn't say instead really, but we can do two things. We can record and see what happens in different parts of the colliculus, or we can electrically stimulate there. Well, so here is a schematic. Here's a colliculus. Okay, this is anterior, this is posterior. So this, obviously, if you remember, this is close to the fovea. And here's medial, and here's lateral. Okay? And what we can do then, we can see where the receptive fields are of these neurons. Okay? And we can plot them out. Here are the receptive fields. Here, number 1, close to the fovea. Number 2 is medial is up, and lateral is down. Now that we've has done this, you can switch over and you can electrically stimulate. Okay? And look what happens. If you electrically stimulate at 1, you get a saccade that moves the eye into the receptive field of that neuron. That set of neurons, I should say. Then if you stimulate in 2, you get saccades that move it to 2. And when you stimulate in 3, it moves to location 3. Now, what more do we need to verify given this situation? How can one believe this hypothesis, if you will, at this stage? What did I tell you about when you electrically stimulate the neurons in the final common paths, like in abducens in the oculomotor nucleus, for example. What did I tell you? That the duration of the stimulation defined the size of the saccade you elicited, right? Now, if that's the case, if that were the case, then this would not mean a damn thing, would it? Because you could take that same location and stimulate longer, and you would get a bigger saccade. So what do you need to do? You need to systematically vary the duration of the high-frequency burst in the colliculus to compare it with what happens when you stimulate in the, say, abducens or oculomotor nuclei. Okay, so let's look at that. Here is that picture I've shown you before. As you increase the duration of the high-frequency burst, you get progressively bigger saccades. So now we do the same thing in the colliculus. And lo and behold, you get something totally different because you have a totally different code in the colliculus. What you get here is that you get, after you exceed a very short time, which is 10 milliseconds, when you don't get a saccade, from 25 to about 120 milliseconds, you get saccades, but they're all the same size, okay? So meaning it's where you stimulate in the colliculus, not how long you stimulate. And then to prove this even further, when you make the stimulation much longer than that, you get a staircase of saccades, each of which is of the same size. Now yet another proof here is that if you stimulate in the anterior tip of the colliculus, remember where the receptor fields are very close to the foveola, then you get a whole array of, like, I think there's something like 14 saccadic eye movements, bang, 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 because the stimulation is on for a long time. So each little saccade is the same size, okay? You just get a staircase of them. So that proves that the coding operation in the colliculus is one that defines the location of a receptive field, and when you stimulate, generates a saccade that moves the eye into that receptive field. All right? So that then, in a nutshell, tells you what the basic principle is of the functioning of the colliculus when it comes to eye movements. And namely, it says a saccade is generated by computing the size and direction of the saccadic vector needed to null the retinal error between the present and intended eye positions. That's just putting it in a slightly different way, saying that, okay, I've decided I'm going to make a saccade to here, bang, like that, okay, just a saccade, I mean. And what you did, that you had an intended location, and by making that eye movement, you null the error. Now the colliculus is really quite accurate. You can make saccades with very small error, okay? Usually the error is roughly at most around 10%, okay? Meaning that when you make very small saccades, it's almost negligible. When you make a large saccade, then maybe there is an error. And when that happens, sometimes what you have to do is to make what is called a corrective saccade. So suppose you are looking here, and an object appears here, all right? And so you say, I intend to go here, and the colliculus fires in that region of the colliculus to generate an eye movement, and say the eye movement goes to here. Then what you have to do is to make a second saccade, like that, to correct for that little error, okay? So sometimes when you make large eye movements, indeed, you make a secondary saccade for the correction, because your accuracy is roughly around 90% correct, okay? Does everybody so far follow the basic principle? Very important to understand the basic principle of the operational characteristics of the superior colliculus. Okay? So again, to repeat, a saccade is generated by computing the size and direction of the saccadic vector needed to null the retinal error between the present and intended eye position. And what you have in the colliculus is a cell that is being activated because a target falls into it. And that activation then generates a signal to the lower parts of the colliculus. We'll talk about that, how that is done, which then results in a saccadic eye movement that nulls that error signal. All right? Very clever, very basic, very beautiful, and almost computer-like at this level. Okay? So what we can do now, we are adding the superior colliculus, the very basics of it so far, to this diagram. And that says that the colliculus then sends a signal to the brain stem, some of it initially to the supranuclear complex, and then from there it sends a signal to the abducens or oculomotor or trochlear nuclei to generate the appropriate eye movements. Okay, so that then is the very basics of the operational characteristics of the superior colliculus. Now what we're going to do next is we are going to examine what the nature of the visual input is, predominantly the superior colliculus, not entirely, but heavily, to generate a saccadic eye movement. So if you remember, I told you that in the retina we make a major distinction between two classes of retinal ganglion cells, the midget and the parasol. The midget project to the parvocellular layers of the colliculus, and the parasol cells project to the magnocellular layers. However, it's also been found that, I mentioned that only briefly, that in the retina there is yet another class of cells that is called, some people have called it the W cells in the cat, other people have called it the coniocellular cells that reside in relatively small numbers compared to the other two, throughout the retina, and project in part to the interlaminar layers of the lateral geniculate nucleus, but it's been found that they also project heavily to the superior colliculus. Now, how do we know this? Well, let me tell you, it's important always to understand the experimental procedures. So let me skip that. Here, just to remind you again, are the connections, all right? This I've shown you several times before, all right? So it says here the interlaminar layers project to the upper portions of the visual cortex, but what I didn't tell you before, that these coniocellular cells also project to the superior colliculus. Okay, so here's the superior colliculus, and here initially we can ask the question of what projects to the colliculus. So how do we find out? Suppose you are in a laboratory where the big question is what kinds of cells project from the retina to the superior colliculus. Think about that for a minute. What kind of experiment would you do? How would you find out? Well, that's quite an interesting question. And so to understand that, I'm going to tell you about a couple of techniques. One technique would be that you could inject, an anatomical technique, you could inject a substance in the colliculus that would be then retrogradely transported to the retina and would light up the cells there. Okay, that would be one approach. Another approach is that you could actually record from the retina itself with microelectrodes, and you could electrically stimulate in the colliculus. And that way would be back-driving cells, and when you found a cell in the retina that is back-driven from the colliculus, you know that that particular cell does project to the colliculus. So that's a very strong technique, because that enables you then to identify the cell type, and also you could, if you do intracellular recording, to label it. All right. So they can do the same thing also, and because I'm going to talk about that as well, is to see what happens in terms of inputs to the colliculus from the cortex. So let's talk about each of those, and that will give us a better sense of what the projections are from the retina and from the cortex to the colliculus. All right, so here's an example of how you do it again. Okay, so we are recording from the retina here, all right, or in different experiment, you can record from the cortex. We can then electrically stimulate here. We can backdrive the cells in the retina, or we can backdrive the cells in the visual cortex. Okay, and then once we do so and are recording from a single cell, we can study to see what it is like. So that is the approach. And if you do that, you come up with some very nice answers. First of all, that these cells are W-like, which means that they're like the corneocellular cells that we have seen in the retina. These are these small cells that are not particularly rapidly conducting, obviously, and are not color selective. And then, if we look at the cells up here, we find that all those cells are complex cells, and they all reside in layer 5. Now, that's already known anatomically, that the cells in layer 5 are the ones that project, many of them that project to the superior colliculus. All right, so that then is the basic procedure. All right, so now we can expand on this basic procedure. All right, what we can do here is we can record from the superior colliculus, and then we can eliminate the input from the cortex by cooling it. All right, that's one approach. So if you do that, you record from a cell here, you drive it by electrical stimulation, by visual stimulation. Okay, we identified that cell in V1 by backdriving it, and then once you cool the visual cortex, you see what happens to that cell in the various layers of the superior colliculus. Well, when you do that, you get a very dramatic and clear-cut, luckily, clear-cut result. The result is the following. If you look at the superficial layers of the colliculus, okay, and look at what happens before you cool, when you cool, and after you cool, the cells keep firing. So when those upper layers, in superficial gray, those cells keep firing. They're not interested in anything that comes down from the cortex. That's because the input from the retina goes to these upper layers of the superior colliculus and goes predominantly to the superficial gray. However, once you get down to the intermediate layers and the lower layers as well, every time you cool, the cell stops firing. Okay, as you can see here, which means that these cells in the intermediate and lower layers of the colliculus are driven by the cortical downflow. They're not driven by the direct input from the retina. So now that we have seen this, one can say, okay, that's fine, but is this really something that happens over time, or is it just something peculiar to a particular experiment that's been run? So what do you do to see if this is really something, a permanent effect? Well, what you can do here is you can take a monkey and you can remove the visual cortex on one side, so the monkey can see fine in half the visual field. And then you can record on either side, the intact side or the affected side in the superior colliculus. Everybody follow that? All right. So if you do that, what you find is this is the intact side, and this is the side where there's no V1, and this was taken months after the area V1 was removed. And here they see this red spot here and this red spot here. That spot is the location after which you no longer could drive any cells visually. So in the intact side, you could drive it to very deep layers of the colliculus. They had responded very well to visual stimuli. But on this side, they stopped responding here. So none of these deeper layers could be activated by visual stimuli, because those deep layers indeed are driven by visual inputs from the downflow from the cortex. All right? So that then is the basic rule of what has happened in the superior colliculus. All right, now let's see. The next big question we can ask is this downflow from the cortex that drives these important intermediate layers which are involved in eye movement generation. To what degree are those corticotactile cells driven by either the parvocellular or the magnocellular layer cells, meaning from the midget cells or the parasol cells? Well, how do you do that? I've already told you about experiments in which what you can do is you can put a recording electrode, in this case in the colliculus, and then you can inactivate either the parvocellular or the magnocellular layers of the lateral geniculate nucleus while you record in the superior colliculus. So what happens is that these cells, then obviously as shown here, project to V1, and then layer 5 cells project down. So what we are going to identify then is what is the nature of these cells in layer 5 that drive down there. So first of all, what this shows is that if you record from, in this case, from 2 cells in the geniculate shown, in 2 cells in the colliculus shown, this is under normal conditions, and this is when the parvocellular layers are blocked. Here's another cell, normal condition, and the magnocellular layers are blocked. So this clearly shows, and this was done with dozens and dozens and dozens of cells, that blocking the parvocellular layers, meaning blocking the midget system, had no effect on the corticotactile driving of cells, whereas magnocellular block eliminated it. So that meant that these cells in layer 5 that project to the superior colliculus are driven exclusively by the parasol system. And indeed then, that was also done, let me go back to that. That was also done in another set of experiments showing that those cells in the cortex in layer 5 that you could drive from the geniculate by backdriving it, if you then blocked the midget or parasol cells, those cells would stop firing. So those are indeed cells that were driven exclusively by the parasol system. So the parasol system plays a very important role in driving the colliculus, but doing it so only through the cortex, not directly. Okay? So to then put this in a sort of easy to remember diagram is to say that when you come to these higher level animals, what happened is that due to encephalization, the cortex gained control over the superior colliculus, excepting the superficial gray. Okay? Everything else in the colliculus is controlled by the cortical downflow. And that, of course, became very important, because when you make a decision as to where you're going to look next, or you're going to identify an object, you have to have information from the cortex itself, which then has to be transferred, in this case to the colliculus, to generate the desired eye movement so that you can analyze in detail what you want to analyze. So that then is the basic layout. And so now what we can do is we can expand some more on this diagram. Namely, once again, let's go through all this. We have the brainstem here, which is a rate code, all right? And then we have a superior colliculus. And that code that we're going to talk about there that I've already described. We can refer to that as the vector code. Okay? And then what we can see here, some of this work I've shown you before, is that we have these three systems from the retina that we talked about now, the midget, the parasol, and the W system, or the corneocellular system, that goes up the cortex. And then the downflow from V1, we now know, is driven exclusively, it seems, by the parasol system. However, remember also that when we go to higher cortical areas, V2, MT, V4, and so on, these areas, some of them get a mixed input from the two systems, like V4 and the temporal lobe. And MT, on the other hand, is dominated by input from the parasol system, but it's not exclusive. So in the end, the colliculus also gets an input from the midget system, but the heaviest input to it is indeed from the parasol system. So that then is what the diagram looks like at this point. And so what we are going to do next time, we're going to expand on this connection, in which we are going to look at some other cortical structures. Namely, I've already referred to that a few times, the frontal eye fields and the medial eye fields, which are regions in the cortex that control eye movements, and that's what we are going to look at in much more detail. Okay, so now I'm ready to summarize what I told you so far. Okay, first of all, there are several classes of eye movements one can distinguish. Those are vergence eye movements and conjugate eye movements. Okay, and the conjugate eye movements come in two major types, which we call the smooth pursuit type and the saccadic type. Okay, then we're going to talk about eye movements as a result of the 6 extraocular muscles that we have. And important to note that these extraocular muscles are such that the fibers in the muscle run the entire length of the muscles, they're not segmented, and that they are innervated by the 3rd, 4th, and 6th cranial nerves. Then we say that the discharge rate in neurons of the final common path is proportional to the angular deviation of the eye. Saccade size is a function of the duration of the high-frequency burst in these neurons. And if you look at up and down eye movements, as I've shown you, if the more the muscles contract that are the inferior recti, the more the eye deviates downwards. And because it's reciprocal, as the eye deviates more and more, the superior rectus is activated less and less. So you have this proportional arrangement. It's a beautiful, as I've shown you, a linear system that defines very accurately the degree of deviation of the eye as a function of the frequency of the neuronal activity. All right, then the superior colliculus codes saccadic vectors whose amplitude and direction is laid out in an orderly fashion and is in register with the visual receptive field. I told you that in the anterior colliculus, the receptive fields are very close to the fovea, and when you go to the back of the colliculus, they're progressively further peripheral, and medial is up and lateral is down. It's a nice orderly arrangement, and you have an arrangement where the visual receptive fields and the motor fields are in register with each other. And every time there's activity in where there's a particular receptive field location, the result is that a saccade will move the eye into the receptive field, thereby nulling the retinal error signal. All right, and then the retinal input to the superior colliculus comes predominantly from W-like cells, but the cortical downflow from V1, which comes from layer 5, is driven by the parasol system. But we should not neglect the fact, I'm not sure, neglect the fact that there are other pathways through which the midget system can also make maybe a more limited contribution in generating eye movements. And as a diagram I had shown you, I demonstrated that the superior colliculus is under cortical control in higher primates. So that then is the essence of what I wanted to cover today. And the next time we are going to talk about, let me see, we're going to talk about the cortical areas involved in saccadic eye movement control, which will then highlight the fact that this incessant activity of us moving the eyes is incredibly elaborate and complicated. There are millions of neurons involved in doing it, and yet it's such an incredible system that is something we never even think about. Luckily, it's a kind of system, partly because of the fact that there's no weighting involved in the muscles, and that the fibers are on the entire length of each of the extraocular muscles. It is a system that one has been fortunate enough to understand reasonably well in how it works in the brain. So that, in essence, is what I wanted to cover today. If any of you have any questions, anything not, I know this is kind of complicated, but I hope that you will understand this reasonably well. And if it's not clear to you, I will be very happy to answer any questions that you might have. Oh, was I reasonably clear then, huh? All right, so next time we're going to do the cortex heavily. I will also show you some interesting movies about that. And I think you're going to find that a really fascinating story of how the cortex has evolved to generate more and more areas that are involved in an incredible number of functions in the generation of eye movements, as well, of course, as in processing visual information. Okay, well, thank you very much.